Okay, good morning, everyone. Welcome to Bioinformatics for Infectious Diseases. And hopefully we have everyone here. If we do not, we're going to be recording this session, and the recording will be available shortly after the session is over. We'll post it on a unlisted link so that those that are participating in this program can all have access to the recorded sessions, and those will be posted on the forum. So let's start by, first of all, reviewing what is this program going to be all about. We're going to be thinking together and working through some of the basic and more advanced methods of bioinformatics analysis, data organization, presentation, and research question answering using this topic or this field of infectious diseases. So we'll focus on the pathogen interacting with the host, and thinking about how do the vectors also fit into that picture. And we've prepared a number of different examples using coronaviruses, Ebola, rhinoviruses, tuberculosis, malaria, and others that we will kind of use as examples. And hopefully you will also help us identify additional interesting topics and projects that we could include in this review. So last time we met and we started talking about how this program is going to work, and we're going to review that in a little bit. But this session is going to be dedicated to next generation sequencing genomics. So we're going to look at what types of sequencing data are there, how is that data stored and organized, and what can we do with those different types of files, with those different data types. So before we begin, let's review what we have completed in our last session. We talked about how to get started, we already had a number of meetings where we reviewed the topics that we will cover and how to find different types of data on public repositories. So we'll continue to kind of expand on that a little bit. And let's see what happened with assignments that we did last time. So last time we talked about how to just kind of get set up and get oriented with all of the different types of resources that we have. And we asked you to complete your profile on the EDU Bioinfo portal. So we'll take a look. Some of you have done a great job at completing your profile. We've asked you to take the free course, Introduction to Bioinformatics. Unfortunately, not everybody had a chance to finish that course, but we do have a number of you that have submitted information in the survey that's included in lesson 1. And then we also asked you to get familiar with the Bioinfo server and Code OmicsLogic Playground, which I saw that a lot of you did check it out and try different things. And so we'll talk a little bit more about how those two different things work so that that experience is fruitful and has results that you can use. So I saw that quite a number of you, and I prepared a few examples of people that have completed their profile. And so what we can see here is Zukain Francis. His profile, he has completed, uploaded a profile image, has some social media links. You can see that he's a participant of the Bioinformatics for Infectious Diseases, and you can see that he is already trying out a few things. Importantly, what I have not seen is the course Introduction to Bioinformatics, but we have some other great profiles. For example, Jeff Ochieng, who's an epidemiologist, researcher, and scientist. So I think a lot of great people that have joined. Benedicta, so welcome all of you, and Nigel Doling. I think Nigel's profile really stood out to me. I saw that he recently started completing all of the information, has already played with some of the coding assignments, and has submitted his profile information on the Introduction to Bioinformatics course. So why is this information important? Because most of you come from very different backgrounds, at least from the submissions that I've seen so far. Some people are just doing this as a part of their coursework, trying to maybe understand what bioinformatics is and how it's used. A lot of you have already advanced experience with bioinformatics, but a very particular aspect of bioinformatics. For example, Nigel his background is in computer-aided drug discovery and discovery of natural product compounds. So obviously this is a little bit different from, let's say, next generation sequencing data, right? So bioinformatics is a broad and dynamic field. A lot of things change, a lot of new topics come up, and the diversity here is really important because I think ultimately in bioinformatics, one of the major trends that we're seeing is multi-omics integration. How can we integrate all of these different types of data together and understand the applications of analysis in a meaningful way? So maybe I can ask now in the chat box, how many of you have started the Introduction to Bioinformatics course? So if you've started the Introduction to Bioinformatics course, you can put number 1. If you have completed the Introduction to Bioinformatics course, please put number 2. Okay, thank you, Benedicta. She has started the Introduction to Bioinformatics course. Rita, Nigel, okay, thank you. Has anyone completed the Introduction to Bioinformatics course? So number 2, okay, Courage, thank you. Number 1. Okay, Kwame, thank you. Okay, so that's also kind of my impression that most of you have started the Introduction to Bioinformatics course, or at least have looked at it, maybe have not yet started. So by the way, before we go on, and I'll kind of, hello, Jeff, I'll kind of take a look at the program itself here before we jump into the course, and I'll show you how you can see progress and evaluate what are the more difficult kind of challenges as we move along. So here on our main Bioinformatics for Infectious Disease Training and Research program, which, by the way, an easy way to get there is to go to events. And this is the first program right here, Bioinformatics for Infectious Diseases, Noguchi. And then you will find here some activity. For example, Francis is running some pipelines. Nigel is trying to run some code. Maybe Kasturi is also trying to do some things. So you can just kind of see updates as they happen on different resources that we discussed. But if you want to understand how is the progress for the whole group, you go to this, to the menu, and there's a tab here called Progress. And here you can see by date. So here you can select the dates. So I selected from midweek last week up until today, so 7 days, and you can see what are the dates for workshops, these interactive sessions, and also what are the averages on different platforms. So here you have the coding in pink and the server averages in gray, and we are yet to see anything in yellow because nobody's really completed the Introduction to Bioinformatics course. And here you can see all of these great profiles that most of you have completed. So those of you that you can see a full name, this is very useful because you immediately recognize who the person is and you can look through their profile. Otherwise, it's not always clear. For example, this person, Badu, we would encourage you to complete your profile. And by the way, we have a full kind of a tutorial on how to complete your profile. So I'll just paste this link into the chat. So anyone who wants to have some guidance on how to complete your profile, that is the link to a full blog post that explains how to do that. Okay, so let's just take a look at that quickly. How to complete your profile right here. So here you'll have a full tutorial, how to update it, where exactly do you go, how do you post the links. I've seen that some of you post links to your social media, but they're not full ones. You're only taking the username. Please put in the full links. This is what a full profile looks like, some tips on how to monitor your activity, etc. Okay, So let's take a look now at what are some of the topics that are covered in the Introduction to Bioinformatics course so that you can anticipate what this will cover. So one of the major trends in bioinformatics, as we've discussed last time, is that from small amounts of targeted data, we are moving into where a lot of data is being available on almost any biomedical research topic, but also including other non-biomedical domains. And this trend is going to accelerate moving into the future, which means that we are exposed to complexity. And this complexity includes different types of data formats, different amount of data available for different areas of a particular topic or domain, and a variety of different types of information that we have to assemble together. And so as we see this big data component, one of the questions that we might ask is, how does this change our understanding of different types of applications of bioinformatics? And so when we speak about applications of bioinformatics, we are seeing, for example, how does this change medicine? How does this change treatment? How does this change diagnostics? What is it that we think we understand, but we are starting to discover more because we have now availability of this big data? And so what's important in all of these is that the field of big data is really something that is very dynamic. And I think that what's important for us in this program is to really understand how can we, in the context of bioinformatics, understand the terminology, the concepts, the different types of structures, and then apply them with an understanding that all of these are going to be changing. So one of the things that we include in these courses are also quizzes to kind of help you test your understanding. And a lot of times I think that when we tend to read articles or publications or maybe magazine articles, right, we kind of glance over them. And when we glance over things, we tend to miss the details. And so we provide these quizzes to help you evaluate how much have you understood, what are the details that you have missed, and hopefully these will be helpful, because as we move into the practical analysis steps, you will realize how small issues, like maybe a wrong file format, maybe the wrong name, maybe a dot or a comma in a certain field, can have a profound effect on your ability to complete some kind of an analysis. So this is something that we really wanted to emphasize and make sure that we have something that you can take away with you after this course. Now, the course completion, this introduction to bioinformatics, is something that is obviously not required, but I do want to mention that similar to this introduction to bioinformatics course, what we will cover today is included in a course called Introduction to Genomics. So we have a number of these introductory courses, especially for those of you that would like to refresh and remind yourself of the different technical and conceptual aspects of bioinformatics. These will be useful. Now, what we have found is that the majority of the people, even though they might already know a lot about these topics, they have positive kind of outcomes from completing this coursework, and a lot of them are also sharing their insights. So if you do complete a course and you are interested and you feel like this was useful, we do ask that you post a response or a feedback or some kind of a rating so that other people can see how they can benefit from these courses. And we have a variety of different types of responses specifically for this Introduction to Bioinformatics course, because even you can see that people who are faculty are finding a surprising amount of depth, and people that have already completed training in bioinformatics are also finding it useful. So hopefully this will be useful for you as well, and not just as a technical step moving forward. Now, what will happen as you complete these different resources? So here's an example of a completed advanced profile. You can see that as the points accumulate, you get to the level of an expert. And what that really is defined by is your ability to deal with a variety of data formats, a variety of methods of analysis, conduct independent work, and move along towards completing a certain type of a section. For example, we have programs on just transcriptomics or just genomics, or more specialized training in a particular application like infectious diseases. So as we move forward, we will kind of identify, and even today I will at the end kind of mention what are the specific collections of courses that we have if you're interested in a specific application. So Sarah Zapico here is a professor at University in Florida, and you can see that she has over 18,000 points. You will also see in your own profile quiz results and how long it takes you to complete quizzes. You also have a personal section for monitoring your progress. And the overall idea behind progress is that you start with theoretical learning, then you move on to practical pipeline applications, and eventually you also add on some practical skills in R and Python, and all of those are different pipelines, but pipelines or code blocks, and you will be able to review your activity under your profile. At the end of this process, we will issue a certificate of completion or a certificate of excellence. The difference between those two is that a certificate of completion just means that you have completed the basic requirements, a certain number of courses, a certain number of points. For people that have gotten over 10,000 points, we are issuing certificates of excellence, where we also specify what does that mean. And that means that you've probably identified an independent project and maybe have completed that project with a presentation and with converting your theoretical knowledge into a practical application. Any question about that? Has anybody had any challenges with completing your profile or understanding the different kinds of resources? Have you had time to navigate through the different resources that we provide? So if anybody has any questions or comments, please go ahead. You can also unmute yourself. We're in a smaller forum today, so this is intended to also be a discussion and not only a lecture. Okay, so if there are no questions or comments, hopefully you did have a chance to look at the different resources and everything is clear. So looking forward to seeing more progress in our next session. So last time we talked about how using bioinformatics, we can study a variety of processes. For example, replication and colonization of the host. Okay, I see a question here from Kasturi. He had a problem in understanding the coding, so we will review that towards the end of the session. Thank you for posting that. Okay, so today we'll kind of continue from where we finished last time when we talked about how using bioinformatics, we can study replication or colonization of host by pathogens. We can monitor what happens to host cells, for example, processes that cells go through and eventually cell death, and also how does that affect organ function, interference, and damage. So how do we describe the process of disease and infection through our understanding of what happens with the host? And we started talking about sequencing as one of the methods to understand and monitor this process. Now, we looked at two types of applications in terms of studying the host, or sorry, the pathogen, targeted amplicon sequencing and whole genome sequencing. And today we want to talk a little bit more about what does that include, what kind of data formats are there, and what can we do with the different data formats. Now, we also talked about how the platform that we're going to be using to understand all of this is going to include a variety of different applications to deal with the different types of data. Now, when you use those different types of applications, it's important to understand what kind of data do you have and what can that data provide you with. Now, some of that data could be proprietary. For example, you've generated some sequencing data and you would like to understand what does this data show. You can also combine your data with publicly available resources, and we'll take a look at which ones, so that we can infer more from the same data that we have. So if we put our data into context, that helps us understand how does this data play a role in completing the picture, or what new information does it give us. And in that context, we'll talk about how sequencing data, for example, can help us study protein function changes by multiple methods. So we can do multiple sequence alignment, or we can identify how that structure folds from an amino acid sequence, or we can study the host response. And so by integrating multiple types of data, we can really understand much more if we have some kind of a targeted portion of that data in our proprietary information. And to understand this, we have collections of data types that we are putting together, and we have already some examples, but we're actually putting together some new resources that include viral genomics and evolution, microbiome and infection, and immune response and biomarkers. So at this point, I wanted to hand over the microphone to Mohit Mazumdar, who is putting together a collection of publicly available resources and data sets on malaria. Mohit, are you there?
Hi, Ilya. Can you hear me all right?
Yeah, I can hear you fine.
Okay. So hello, everyone. Yes, so as Ilya was telling, that we are kind of compiling a document where we have looked for the data sets that are available publicly. And we wanted to share it with all of you so that you can also see the resources that we have compiled, and maybe also can contribute to some of the resources that you are aware of. So Ilya, you have to make me host. I think I have another login.
1 second.
Thank you. Okay, so let me share my screen, and I'll be able to show you the document that we have been compiling. So in the sessions, you started with looking at some of the data sets. We did it in our last webinar. We looked into NCBI. So NCBI is one of the major repositories where we find a lot of important data and publicly available data that are getting submitted every day. So other than NCBI, we wanted to look for other places that have other research groups, other organizations, and other portals where you will find data. So one of the portal which has quite enriched information about this particular parasite is ENA, so European Nucleotide Archive. So if we go to the browser, and then if I write Plasmodium and do a search, so I'll find a lot of information here about the assembly, about the sequence, and about the transcriptome. And then you will have the SR experiments, and then there are like these studies that have all the information about that individual experiment or the research. So you will find a lot of information here in that ENA browser. So other than that, there are other resources as well, which I think many of you might have already explored, because some of these resources are quite important for malarial research. So one of which is PlasmoDB, and I found a lot of citation with this organization, because there's a lot of valuable information that is there compiled in PlasmoDB. So it's a funding based on the Wellcome Trust and Bill and Melinda Gates Foundation that started this and being able to do this at such a larger level. So in here they have also like tools to do your own stuff, but I think that has limited options because you cannot download and upload data. So we looked into this and we found that there is a lot of information on transcriptomics and genomic data sets that could be seen here, well annotated, one place. Another important place, again, this is like a very basic place that we all go to download the data, to download the genome or the transcriptome, so the Sanger data set. So here you will find the process UTR regions for the Plasmodium falciparum parasite. And then we looked into another consortium or a group, Malarial Host Parasite Interaction Center. So they have very interesting case studies that has a lot of multi-omics data. So, um, so we have kept a kind of link here. So if you go to this link, you will find experiments, and then you will find the data sets that are publicly they have made available for you to look at and understand some of the things about data integration. So they looked into the system biology approach. So uh, mm, there are multiple technologies that they're combining to come up with solutions or strategies. So you will see here a lot of resources that are associated with each experiment of different kind. So, and also the links to download these where they are uploaded. So gives you a good perspective of what all is there. And then after this, sorry, we kind of trying to describe the application of these multiomics technology and why metagenomics and how metagenomics can be used to understand the parasite. So this is from a review. So we have compiled these from different resources like reviews, research publications, and try to put it in one place where you can see the application and also find relevant data sets that you can apply and learn in this research training. So again, then it talks about the evolutionary relationship that we will be covering in our infectious disease session, and then talks about the importance and what how it can be applied to the the research problem and what we are looking for malarial research. And then it describes about the transcriptomics and application of gene expression and how it can be related with clustering and what insights it can give. So there's some examples that we wanted to put together so you can see that how it can be applied and have been applied in different publications. So you'll also find the citation. So it's a documentation in preparation, and we wanted to share it with you at this early level so that everyone of us can contribute to this document and we can then share it with everyone else out there so that we come up with a contribution and we can go back and see that these are the resources at one places and we can click on those specific resources. So from sequence to we move on to structural part. So how do we find structural data? And that you can we started looking at the data that are available on protein data bank, looked into some of the structures and available data sets, available structures that are there with plasmodium. Surprisingly, protein data bank in itself has crossed 1 terabyte of structural data, which is amazing. So there's a lot of information, structural information that is there, and we're kind of preparing a case study on our PC PFCRT gene, and we'll be preparing this as well. So very important mutation for chloroquine resistance transporter gene. And yeah, this is it. So the plan is to share it with you all, and you can comment and you can add your own data sets which you or the resources that you have gone through or know of. So it will be great. So thank you. I'll just pass it on back to Ilya and look into comments if you have shared anything.
Thank you, Mohit.
So as you can see, in the case of malaria, which I see that many of you are interested in, we go from an overview of available public domain data sets, curated data sets that you will find as projects on the educational portal. You will find how the same type of an infection could be described with multiple types of data types, and we can then integrate those data types to really understand how do they describe what we are interested in. So for example, as Mohit pointed out, there have been publicly available studies on how malaria spreads and what distinct subgroups it forms. We also will look at the relationship between sequence and function in many cases. And what our objective is in the end is to also provide you with a resource and with an ability to then discover your own data sets and structure your analysis accordingly. So to do that, we have, and I've pasted in the chat box, a link for your submission. Jeff requested this as well. So if you look on the first, so on the forum right here, you will find the first one is a post about project submission. So this is to help guide you submit additional resources. For example, you've seen some general databases and case studies that Mohit has pointed out. If you have a data set of your own and you have maybe a particular interest, this will help you submit that for our review and maybe for other people to join your project as a collaborator as well. So here you need to fill out your first, last name, email, city, etc. But then here we have several different sections. Company name, which is your university, project title, so something that you would name your publication in the end, problem statement, project summary, how do you analyze, what are the data sets, what are the specific types of insights you're looking for, link to raw data, and reference publication. So if you could take a look at the forum as we go through the program. The earlier you start putting together a draft, this is a dynamic form that you can change over the course of the full program. So you will also see here some introductory videos. In these introductory videos, we have a session dedicated to developing your own project data set or formulating your project idea. Right here, this one that was posted by Beepsa. How to find omics data and how to structure that into a project. Any questions about this so far? Or any comments? Okay, so again, please put your comments or questions in the chat and we'll be able to address those. So as I mentioned, this is a way for you to submit your data. You've seen a resource. If you want to have access to that document, please connect with Mohit at pine.bio, or you can put it on the forum. We'll start a discussion there. Any other types of repositories that you think are important to include, we'll start looking at them. So let's talk about today's topic, which is next generation sequencing genomics. So obviously, sequencing has been such a prominent data type in the context of infectious diseases. We need to understand what are the basics and what are the data formats. And I know that for some of you, this is going to be repetitive. You probably know a lot about this. A question from Kwame real quick. Should the project be related to only those pathogens you've listed? No, this is your own project. So whatever topic you are interested in, whatever pathogen or host or any kind of a infectious disease related topic that you have, please do it around those topics. So what does sequencing do? The machine itself captures visual information that allows us to recognize nucleotides. What are nucleotides? Nucleotides are, in the context of DNA, molecules that are made up of sugar called deoxyribose and a nitrogen-containing base and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds that form between the sugar of one nucleotide and the phosphate of the next, resulting in an alternating sugar-phosphate backbone. So the nucleotides that are joined together are called base pairs. The base pairing connects the two strands of DNA. When referring to the number of base pairs contained in a sequence of DNA, we are referring to connected bases. So when we speak about the sequencing that helps us differentiate between adenine, guanine, thymine, and cytosine, we're always going to be talking about DNA. Even if it's RNA, it is converted to cDNA. And so this visual signal is detected with a certain accuracy. And so we'll talk about that in a little bit. Now, these nucleotides form codons. So essentially, the genome of any organism is organized into what we call genes. And those are built for translation. So essentially, each gene is like a recipe for a certain protein. It encodes instructions from which a protein can be made, which is the gene sequence. Proteins are the product of amino acids, which are encoded by trinucleotide codons. So a codon is a sequence of 3 DNA or RNA nucleotides that correspond with a specific amino acid or stop signal during the protein synthesis. Each codon corresponds to a single amino acid or stop signal, and the full set of codons forms the genetic code. Once the right place on the DNA is copied over, it is prepared for translation by forming a messenger RNA or mRNA that is exported from the nucleus and the cytoplasm to be produced. So RNA polymerase is what makes the RNA from DNA, and the accuracy of conversion is also required for that RNA polymerase. So what are the types of DNA variation that we're looking for? Sometimes we have a mutation. That mutation is a change in a single nucleotide, and that nucleotide can change a number of different things. So to really understand the impact of different mutations or variants, we can talk about a number of different examples that actually happen, right? So everybody, I am assuming, is familiar with Pikachu. So you can see here that the regular Pikachu looks like this. Sometimes the Pikachu changes its phenotype, right? So here we can see blue eyes. That doesn't mean that the variant is a mutation. It's really just a variant, right? So it's the same Pikachu, does the same thing. It just looks a little bit different. Sometimes it can change something about its function, and that could be harmful or that could be neutral. So for example, here included with everything else, he also has wings, right? So it's not necessarily harmful to anything. It's just a novel phenotype, but sometimes it actually changes some function. And that is really what we talk about when we say pathogenic mutation, is a mutation that changes function in a harmful way. And so the types of mutations, and again, I know that many of you are probably familiar with this, are several different kinds. For example, missense mutation is a change in one DNA base pair that results in the substitution of one amino acid for another in the protein made by a gene. Nonsense mutation. A nonsense mutation is a change in one DNA base pair. Instead of substituting one amino acid for another, the altered DNA sequence prematurely signals the cells to stop building a protein. This type of mutation results in a shortened protein that may function improperly or not at all. A frameshift mutation, which is a type of mutation that occurs when the addition or loss of DNA bases changes the gene's reading frame. A reading frame consists of a group of 3 bases that each code for one amino acid. A frameshift mutation shifts the grouping of these bases and changes the code for amino acids. The resulting protein is usually non-functional. Insertions, deletions, and duplications can all be frameshift mutations. So sometimes these changes at a single nucleotide can have very different impacts on what we observe in the final code. So as a result, we can see how some of these could be a change in a single amino acid, and that can significantly change the function, right? So here we can see how one single mutation changes one amino acid, and that amino acid changes the phenotype, or how the final product looks like. Now, the genes also have a very particular structure, because sometimes we think that a mutation can only happen within the gene. But in reality, there are upstream regulatory sequences and downstream regulatory sequences. And so when we think about mutations appearing in a genome, we oftentimes are only focused on finding these amino acid changing mutations. And as we know today, the regulatory sequences that have an impact on whether the gene is enhanced or silenced, and in what quantities that is produced, could also be affected by mutations. And we know that there are even intron variations that we observe that have an impact on ultimately what is going on. So it used to be that genomics was reduced to only gene sequence variation. Today, genomics is looking for variation across the whole genome, and the interaction between minor frequency mutations, major frequency mutations, whole inclusions, rearrangements, and insertions can play a big role. So you can see here that you have these splicing control variants that could have a major impact. Now, there is also a variety of types of sequences, for example, in the genome. So you can see here about, so I think this is for the human genome, there are transposons and fragments, which constitute about 45% of the human genome. You have a lot of defective viral sequences and fragments, full virus sequences about 0.1%, nuclear mitochondrial DNA, protein coding genes only compose about 1% of the full human DNA. And the length of the genome varies significantly between different organisms. So this is, I assume, for most of you is familiar material, so we're not going to spend too much time on this, but are there any questions? So about what are we actually sequencing? What is the material, the biological material that we are looking at, and what can we find in this biological material? Any questions? Okay, so let's talk about the process of sequencing a little bit. When we look at sequencing and we speak about generating digital data from this genetic material that we've collected, how can we inspect different types of information from the sequencing data? So when we talk about Illumina sequencing, which is the predominant kind of most popular way of sequencing today, we're talking about sequencing of short reads. Those short reads could be generated by preparing the correct library. And that library could be prepared for whole exome sequencing, whole genome sequencing, which, for example, when we talk about the human genome, would constitute about 20 to 25,000 genes. Or if we talk about the whole genome, we're talking about 3.2 billion base pairs in the human genome. So we're talking about basically a balance between cost and coverage. Now we'll talk today a little bit more about coverage and the impact of coverage on our ability to detect certain type of information in the sequencing data. But practically speaking, we are talking about a difference between megabytes and gigabytes in terms of data. So when we look at average exome size of the human, it's about 45 megabytes. You can see here from a whole exome sequencing, we're talking about 8 gigabytes, right? And so that 8 gigabytes is when we have high coverage of all of the exons in the human genome. When we're talking about the whole genome sequence of the full human genome, we're talking about 120 gigabytes, and that is a much lower coverage of all the positions on the genome, right? So even though we have this difference, if we were to sequence just in the same coverage the human exome, it would be a much smaller data set than we have for the full genome. And so today, when we think about also pathogens, we can amplify genomic sequences of particular viruses, bacteria. We can separate them from the biological material, but ultimately it's a trade-off between sequencing costs and coverage. And the coverage determines how accurately can we determine a particular position. And that means that we have a sensitivity here at play. So there are also many different types of sequencers that are not just Illumina sequencing, and those could be used for a variety of applications. For example, if we talk about Oxford Nanopore, which is very affordable and produces long sequencing reads, that could be a very beneficial way to detect full genomes and assemble short reads into genomes. A lot of times that's also useful to detect very high frequency mutations, but many times if we want to get greater detail, that's going to be limiting because the accuracy of each position is not so great. When we talk about other types of sequencers, they range in a number of different sequence lengths. So we can have reads of anywhere between 15,000 bases to over 100,000 bases if we talk about the Pacific Biosciences or PacBio sequencing, right? So really long sequence reads. And those, for example, are used today when the areas of the human genome with many repeats are being used. A lot of times, these types of sequencing approaches could be much more expensive, as we see with PacBio, much more expensive than Illumina, or much cheaper. For example, Oxford Nanopore, much cheaper. And so a lot of times we're also seeing an integration between different methods, right? So one application of the same genomic material could be done with a PacBio or an Oxford Nanopore to provide us with the length. Some of it could be amplified using Illumina sequencing, and that provides us with coverage. And so the important part here is that we are also limited by the amount of biological material, because every time you do sequencing, that material is spent. So a big part of the sequencing process is PCR amplification. So if we talk about Illumina sequencing, the process goes the following way. We have genomic DNA, and again, if this is RNA, that is converted to cDNA. We have the process of cutting it up, the material itself, cutting it up into very short pieces, then adding adapters to create a sequencing library, applying it to a flow cell, then amplifying through bridge PCR amplification the genetic material, and then the sequencing part, which again looks like this. It is a visual signal that is converted by the machine into digital signal, right? So this is the process of getting this digital information from biological material. Once that process is done, there are three important data types that we're going to be talking about, and hopefully this will clarify how we can use different types of analysis methods, also based on the different types of data or different formats of data. So does anyone know what is the difference between FASTA and FASTQ files? Okay, anyone at all? Do you know what is the difference between FASTA and FASTQ files? Okay, thank you. Joelna is saying FASTQ has the quality scores. Courage is saying FASTQ has quality scores. Francis is saying FASTA primarily produced by Sanger sequencing. Bright is saying FASTQ files come with quality scores while FASTA has none, right? And FASTQ by Illumina. Okay, so great answers here. Let's take a look, right? So exactly, Anita is also mentioning reads. So what we are looking at are different types of storing that information. The information ultimately could come from sequence reads in both cases. So for example, here you see reads in a FASTA format, here you see reads in a FASTQ format, right? Even though oftentimes we think of FASTA files storing, for example, reference genomes or a full chromosome or something like that. And we also have a third type of file, which is the genome transfer format, or GTF, or oftentimes GFF. So let's take a look at these different types of data. Essentially, all of these are text files, right? So they are ASCII code, and they have sequence. So for example, in this FASTA, we have the name of the sequence, the NCBI ID, and then we have a description, Spodoptera frugiperta isolate SF9 scaffold 12. Scaffold means it's like a chromosome, so essentially this is a short reads assembled into a longer sequence, whole genome shotgun sequence. So you can see that this is an assembly. Here you can see a virus, rabies virus, complete genome, right? So these are NCBI codes. This is a reference that you can then search on NCBI and find. And here we have the FASTQ file. So here we have individual reads. For example, we can see that this is the ID of the read connected to typically the archive of the read, which would be the SRA, maybe the name of the full file. You can see here the length. So let's take a look a little bit closer. Here we have the length is 160. We have the actual read sequence, right? And we also have the quality. So here you have the read, here you have the quality of each position. Now, based on this quality, we can determine whether we can use the full sequence or we should trim it based on poor quality. And we can also understand whether our overall quality is good. So there are a number of different outputs that we will look at in shortly that allow us to deal with different levels of quality. Now, what is alignment, right? Those reads, let's say 160 base pairs long, have to be aligned to some reference genome. And so when we do that alignment, we use another file format to store the alignment results, which is a BAM or a SAM file. Essentially, what that means is that we have individual reads. Those reads have to be identically placed on the reference. And then we have a collection of reads, and they might have some overlap, they might have less overlap, and they might have a complete consensus. In other words, they agree completely on the full sequence, or there might be some variation that we think might be as a result of different types of technical or biological differences. So to kind of illustrate how this actually works, we have to look at an example. How does this look when we align the reads? So to show you how those look, I'm going to ask Harpreet to show us an example of a demo pipeline with an assembly with how those reads map onto the reference genome.
Thank you, Ilya. So just let me share my screen. Okay, so how we can actually visualize our reference genome, visualize alignment of the reads on a reference genome. So one of the important point here that we should have raw sequencing reads in the FASTQ file, and we should have reference genome must be our FASTA in the FASTA format. So for that, let me share two files with you so that you can proceed with me and just go with that. So first of all, I'm sharing one file. This is what our FastQ file. So here we have actually a run table, SRA run table. So for this, I have taken an example from the Ebola. So here we have FastQ files against the BP40 protein. So I'm sharing this example file with you. This is our SRA run table. So this is SRA run table where we have FastQ files for the reads that we have obtained already. I have taken this data from the NCBI SRA. So these reads belong to BP40 protein of the Ebola. Then we have another table that I'm going to share, that is our reference genome. So let me just share this as well. So download both the files. So once you're able to download these files, let me know and just type yes and no. So let's go over our
One second.
Okay.
Okay, so these are, the last link is an SVL, which is a reference of a file that is already on the platform. And as you can see, Harpreet, if you could go back to the SVL file.
Yes. So these are the
You can
links
see
for the
that one of them is a FASTA file, another one is a GFF. So one is the reference genome, another one is the organization of that genome into different parts.
So once you're able to do that, just let me know so that I can proceed. So now you can go to the area of analysis. So just type your answer yes if you're able to download both the files. So does any one of you, okay, that's great. So the reference file is actually FASTA file, that is in the FASTA format. And the other file that we have, that is GTF file for the annotation purpose. So once you are there, just go the first section, that is RNA-seq chip parallel analysis of NGS or microarray data. If you click on this, as here we are not actually selecting any model genome, so we have to go to this R genome GTF. So then you have to select which kind of input file you have. Right here we have raw sequencing read, so its format must be FASTQ. So then we have to select whether they are single reads, single end reads, or the pair end read. So the example that I have provided you, it contains pair end reads. So I can show you which files I have taken. These first two, sorry. So these first two files that I have taken, that is Ebola BP40 bond RNA. So I have taken these two samples in this example, and they are pair and read. So how you could know, even if you open that file? So otherwise, if you click on this sample, so you will understand what is this sample it contains. So like it layout is parent. So here I'm selecting parent. So for both the samples, we have parent reads. Then if you go to the next, once you have selected these things, first type of reference genome, that is R genome GTF. Then format of NGS data, that is FASTQ. And then this is the single on the pair and reads, the pair and reads. Then next go to the file upload. So here first we have to upload our input file, that is the FASTQ file. So these FASTQ files we have loaded in the form of just simple SRA run table. So under here, NCBI run table, just upload your example file, that is example.txt. Once you upload this file, the next is you have to select the organism. So under the organism, I have already provided you one txt file. So we are not loading as such. So here we are going to add those links, add files from the links. So for the organism, here we have to upload that reference genome links for the reference genome file. So I am again. So I am copying these SVL links and pasting first here. This is for the FASTA file, that is reference genome. Other one for the GTF file. So once you're able to do that, then next go to the group selection and just cancel it. Cancel no groups, as here our aim is only to visualize the alignment of these reads on a reference genome. So we are canceling this cancel no groups. The next step that we are going to run, that is start. As we have already provided, uploaded no files, now we can click on OK. Then here we can skip these pre-processing step, as here our aim is only to visualize alignment of reads on reference genome. So the next is under the which tool that we are going to use to map our reads on reference genome. So I am selecting this Butai 2G. So once you select that, the next step is save. Then next option that is automatically get highlighted, that is visualization. Click on OK. Then click on end. Now you can name your pipeline. So I am giving Ebola demo pipeline, demo visualization. So you can name, once you name it, then go to the save. And then you have to run this pipeline. You need to click on this run pipeline. So it will take 5 to 10 minutes. So just let me know how many of you able to complete this pipeline, at least able to complete these steps. Okay, so I can again.
Maybe 1 second. Maybe you can actually just scroll down so you can see all of your pipeline steps.
So once you upload your file, the next step, just click on start. Then choose mapping of your reads on a reference genome. For that, you choose Butai 2G and then choose visualization and finally end. So does any one of you able to complete this pipeline? I mean, pipeline obviously will take a bit time. So just able to complete these steps. Does any one of you able to complete this pipeline?
Should we repeat?
Okay. Okay, so let me again just show you the steps. First, if you go to this create RNA seq, sorry. So let me go to just area of analysis and select this pipeline RNA seq. So from here you have to select first the third option, that is R genome GTF. So as we are selecting new reference genome, so just do that, and then in the format of NGS data, as we are uploading FASTQ file, so choose this option, then just go to the from the single end and pair end, just choose pair end reads. So then next click on file upload. Just how many of you able to go to this step? Just let me know that so that I can proceed accordingly. Okay, so I can paste again example pipeline. So this is the example. We have two samples. So for that I have generated SRA run table. So that sRNA table is named as example.txt. So if we go to, we have to upload our file. So here under NCBI run table upload, just upload this file, example file. Once you able to complete it 100%, then next select organism, which is here shown, third step. So does any one of you able to complete these steps? First, able to choose the option. The next is file upload and then go to the organism. Okay. So apart from Courage, who is else who able to complete these steps? Or if you are facing any issue, you can also ask me. So the next step, we have to provide or this reference genome for this organism. Here our main organism that we are doing for this analysis, that is Ebola. So then we have under here, file from the links, just click add more. So here first we are uploading this SVL, copying this SVL link. So first I am just the SVL links, reference genome underscore SVL underscore file dot links. So from here I am just copying first the FASTA file link, SVL link for the FASTA file, that is for the reference genome of the Ebola, then the GTF file. So once you do that, then go to the group selection. So you can just cancel this, cancel no group. So now your both input file as well as the reference genome get uploaded. The next step, we have to run this pipeline, right? So for that, first need to click on start. Then just choose this option under the mapping on genome. Just choose this option Butai 2G. So we are choosing this algorithm for mapping or the alignment of the reads on a reference genome. Then we just, after choosing the alignment, next the option that we are choosing also automatically get highlighted, that is visualization. Once you do that, just click on end. Now you can name this. So before naming, I'm just asking how many of you able to complete this pipeline. Does any one of you able to complete this pipeline? Okay. So now we can name it Ebola. Next, you can click on My Pipelines. As actually it will take a bit time. So meanwhile, I have already run one pipeline, so I can share its link with you as well. It will take 10 minutes at least. So let me share this link with you where pipeline already completed.
Okay, thank you, Harpreet. And hopefully everyone is able to run this pipeline. Now, what you will see is that this pipeline is described in detail in some of the example projects and courses that we have included. So we will take a look at this in just a second in the results. But let me briefly, before we jump into the results of this pipeline, show you where you can see whether you have created your pipeline correctly. Okay, so I'll just take over this sharing of the screen. Okay, so all of the activity related to pipelines is going to be right here on your organization page for bioinformatics for infectious diseases. So as we scroll down, we can see different types of pipelines that are being run. A lot of times you will not see that progress directly on the pipeline page, right? So if you're on your pipeline page and it says right here started, or maybe even it's black and just says prepared, that means that you have to click on run pipeline. And another important point here is that the same types of analyses that we are doing are available in several different sections. So today, Harpreet has shown you how to do it in the RNA-seq section. You can make the exact same pipeline in the variant calling section. So for example, here, I am looking at the variant calling pipeline. And here you could also do visualization. For example, bow tie and visualization. You probably recognize the same names, right? So once you learn this logic, once you kind of go through several of these examples, you will see the logic. The logic is that once you understand what is mapping, you can select the right one. And once you understand how after your selection, your next options are going to highlight, you can start building many different pipelines. So as you go through these different examples, you will start seeing what are the important things. So for example, we can see here someone, Zukang Francis, right, is running this pipeline. So let's take a look. what could be, so here you have the visualization link, and here you can also open the output files and you can see that there's nothing here, right? And so for example, if we click on visualization, you can see that there's a problem. Something was not done correctly. So let's try to troubleshoot and understand what was not done correctly. Okay, so here you can see the example file, okay, that was uploaded. And this means that the way the file was uploaded was actually incorrect. So it matters where you upload the file. So here the file is correct itself, the file itself is correct, but where it was uploaded is incorrect. So I'm just going to, and again, this is a good example because Harpreet was showing how to do this in the RNA-seq section, but Francis was trying to do it in another section called mutation variant. So here you can see RNA-seq, here you can see mutation variant, a different place. So how do we do it in the mutation variant? In the mutation variant, you also have to make the same selections. So here, instead of R model genome GTF, you select R genome GTF. Here you select parent, and here you select FASTQ. After you do that, file upload. You don't select the file from here, which is what I think Francis did. I think you have to run NCBI run table upload, right, Harpreet?
Yes, Celia.
Okay, so here we have this example file. Okay, why are we doing this from NCBI? Because if you look at the actual file, this file doesn't give you, it's not a FASTQ file, right? It's a TXT file. And here it only has a reference to the SRA. So here you can see SRA download. SRA is a sequence read archive. So it contains inside a number of different FASTQ files. And those are dependent on the link. Okay, what is the difference between R model GTF and the R genome GTF? R model genome GTF are the genomes that are already uploaded and indexed on the platform. So these include the human genome, the mouse genome, the Drosophila genome, so some of the model genomes that typically are used. We have here Ebola genome, right? The Ebola genome is not one of those model genomes. And so that is just a genome. What is the GTF versus non-GTF? GTF means you are uploading a GTF file. It's a genome transfer format, so it's annotation of the genomic elements that are available for that genome. For example, the genes. In the case of human genome, it's the exons and the introns, things like that. Okay, so when you don't have a model genome, you have to upload your own file for the genome. If you chose the model genome, you don't have to deal with the genome. You just select from a dropdown. Okay, so now here, there are two ways of uploading this reference that Harpreet mentioned. One way, because you only have two files, is you can use the actual links. The other way is you might have a full SVL file, right? So here you can also remove them. And here you can choose URL bulk uploads. And let me quickly find this just to show you. Okay, so this is an SVL file. And the SVL file will contain links to those files that we have used for the reference. So I'm not going to do this, okay? But this is just to clarify what are the different file options. So Harpreet, I'm going to transfer it back to you so you can show the results. and there's a question here. Can we upload a TXT file with all the links? Yes, but you have to specify which link is for the reference genome and which link is for the actual files that you have with reads, with the sequences in them. Harpreet, would you like to share your screen and show the results?
Yeah, yes. So let me just paste this link in the chat as well, because this pipeline is already completed. I don't know whether your pipeline completed or not, but it will take sometimes 10 minutes or more. So let me just post this link in chat as well, so that you can visualize your results. So here, if you open this link, you will direct it to this interface. Under here, the genome visualization, if you just click on this visualization, this is blue color. If you click on that, you will direct it to this page. So from here, you can actually visualize your BAM file alignment. So just click on this. Or you can click on any single file as well. So once you are here, this is your reference genome. So now you can actually visualize different section. You can select that. And so these are your reads that are actually aligned against this reference genome. So we can zoom in. So here you can select that. Now these are the reads. Now if you want to look at what these reads contain, so if you just click on one read, this is your reference genome sequence. So if you even zoom more, so you can actually sequence of this reference genome. So then if you just click on these reads, any of these reads, you will get detail regarding each of this detail, like what's its sequence, what's its quality, and how much length it has. Like then you can actually look at all the complete detail here. Sequence length, for example, here under this sequence length, it is 101 nucleotide long. Then you will get even the quality score, and this is what would be the sequence. So you will get all that all complete detail regarding each of these reads. So this is the one thing.
Hello.
Okay.
Hello.
Then okay, so you are saying my pipeline is done. When I click on visualization, the open in error. So I don't know. Can you share? I mean, we can look at the link. Look at your pipeline.
So let's do this. I think that, for the time being, we need to look into the step-by-step instructions, and those step-by-step instructions are provided in an online course that we have. So after the session is over, we can take a look at Courage's pipeline. But just to complete this session so that everybody else who did have a good result can try out different things, we'll move on. To summarize about this result here, right? We see here, maybe you can also click on the coverage and snip.
Yeah.
In the top left. No. Where you have the selection of NGS category for this track, for the tracks. No, on the left. That's the right, on the left. The left. Yeah, where you have the selection, you have BAM alignment and then coverage and snip all the way to the left.
Left.
Keep moving left, even more left. Yeah, there, you see? No, don't.
Okay, sorry, this.
Exactly. Yes. So you can see here the yellow bar, for example. The yellow bar, you can see it on the reads on top, and you can see it on the coverage on the bottom. So essentially, when you have coverage and SNP, what does SNP stand for? SNP stands for single nucleotide polymorphisms. So those are the variants that are clear in all of our reads. So if you go through the whole sequence alignment that you have in the result, you might be interested in individual reads. But most likely, you're not going to be interested in seeing all of the data. You're likely to only focus on the differences from the reference genome. And so those are highlighted for you in this visualization so that you can easily spot them for a long sequence. Now, in this example, we have a very short genome. This genome is about 18,000 nucleotides long, and therefore we can easily see where we have our data, where we have the variants, etc. If you go to the top, you have a drop-down. Yes, right there in the drop-down. So if you have just a single genome like this one, it will just be one thing. But if you have, for example, the human genome and you have all of the chromosomes, all of the individual chromosomes will be in that dropdown, and you will be able to see individual results. So as you go through these examples, try to spend some time to look at the input data, the reference genome, the different types of outputs, and what outputs actually mean. What do they tell you? What is the information included in this example? So to just kind of summarize, thank you, Harpreet. I'm going to take over.
Thank you, Ilya.
Okay, are there any questions about what we did that are not technical? So in other words, I know that some of your pipelines did not work well, and we will take a look at those in a second. Did anyone have any questions about what we did or what does this output mean? Okay, so if you do have those questions, please put them in the link. So to summarize, let's review what we did today. We took a look at how to work with different types of input data. For example, we can take genomic sequencing data as FASTQ files. We used visualization, but here you can see in the DNA variant calling section, other types of methods. For example, pre-processing, mapping and alignment, variant calling, differential variant calling, annotation, and post-processing. Here are a couple of example pipelines that you can make. One that we did today was with visualization, just seeing what's in your data. Another one is an example of somatic mutation variant calling, which is relevant, for example, in cancer, where you have one normal, one tumor sample. In the example of visualization, we can typically find important variation. So this example that you see on my screen shows you how the individual variants are highlighted here by these dots, markings, and you can also see what's inside every individual read. A lot of times, you want to combine this information. For example, information about coverage, how many reads you have, and information about what type of variation is observed in how many of the reads. So here, we will see how this information is translated eventually into some important phenotype. As an example, in cancer, we see here a number of mutations. Those mutations can have numbers of reads that include those mutations. They can have distribution for different types of proteins. Okay, a question from Francis. Can we use the same process to detect multiple nucleotide polymorphisms? So as you see in the example that Harpreet just showed, it wasn't just one reported polymorphism, it was multiple. Now the question is, what do you do with that information? Briefly, before we finish, I wanted to show you another example that is not related to visualization, but that is related to an example that we will ask you to complete before the next session. So this is an example now under metagenomics. So a lot of times you have a variant because you're looking at sequencing of one particular genome. In many cases, you don't know which genome you're looking for. So here we had an example of mapping to a reference database, to a reference genome. We can also map to a reference database. So how would you do that? Here under metagenomics microbiome, I'm just going to show you here an example. In this example, we take Spotoptera frugiperta samples, which are samples from a cell line from a moth, a fall army moth, that is typically used for production of vaccines and different types of biologics. A lot of times you're looking for contamination. You don't know what genomes might be present there. And so it's logical to, instead of mapping on a single reference genome, to map on a whole database of genomes. So in this brief example, I'll just show you what that pipeline would look like. I'm just going to click on Start. Again, I'm going to use Bowtie, the same method that we already talked about. And then instead of looking at how many reads map onto a particular genome, I'm going to see how many reads map onto a whole database of genomes. And as a result, I am going to get a table that shows me what are the abundant genomes that I have found. Okay, and so this was mapped on a database of all viruses from NCBI virus. So from NCBI virus, we can download a whole database of viruses, and those viruses are going to reveal whether there is something dangerous. Okay, so you can select here virus genomes as a database, and as a result, you get the count of how many genomes were identified. And these genomes can be harmful. Some of them are harmful, some of them are non-harmful, depending on whether they're human viruses or non-human viruses. So this project example is shown in this project called Spotoptera frugiperta fall armyworm. So for our next session, we're going to ask you to work on this project because it illustrates, gives you some practical guides on how to run a pipeline and how to visualize your data, how to map it on a reference database, also how to map it onto a reference genome and get the not map reads. So it involves a little bit more detail than what we were able to cover in today's session, but we hope that it's going to be useful as you are starting to look into presence of different virus genomes, variation in virus genomes, and differences of sequencing, pair-end reads, single-end reads, etc. So this tutorial will allow you to go through some of those examples. So for our next session, we have several different things that we would like to ask you to work on. We have this project, Insect Cell Lines and Biologics. We also have a collection of data sets that Mohit introduced today related to malaria. If you have any ideas for additional things that we should include there, please email us. And we have a number of introductory courses. So you can go to the program page, and on the program page, you will find the coursework that is included in this session. So you will see here how to do genomic data analysis, metagenomic data analysis, and transcriptomic data analysis. So here we have Introduction to Genomics. Here we have Metagenomics 1 and Transcriptomics 1. Right. So these are some of the courses that you should start working on. And then we will go on to discuss those in our next session. So the genomics courses, we'll talk about Introduction to Genomics, which includes basis of variation, and the significance of variation in different examples. We have a whole section of courses on transcriptomics, which we will get to in a little while. And we kind of briefly talked about metagenomics, because a lot of times we don't know what we're sequencing. Our question is, what is prominent in what we are sequencing? So in our next session, we will talk about how to use this kind of data to start asking questions. So what are some of the analytical challenges? How can we look for specific patterns, and what can those patterns reveal about the data that we have? So again, I know that it took us a little bit longer today. Hopefully in our next time, we are also looking to improve the interaction. So hopefully before we meet next time, you could already complete some of the materials on your own independently, so that when we meet, we can focus on the technical challenges, how to resolve them, or the specific research questions that you might have about the data. So now, I think this was all the content that we wanted to cover in today's session. So for those of you that have any technical issues that had the issue running the visualization pipeline, please stay on. And for the rest of you, thank you for joining today. We hope to see you again next time. Our next session is going to be on February 5th. Harpreet, I'm going to pass it on to you to troubleshoot the pipeline from Courage.
Okay, Leah.
Could you share your screen and take a look at his pipeline?
Yes, I'm just copying his. Clicking on. This is his pipeline. Let me first look at.
Let's start by looking at the input data always.
I think his data is not getting.
You have the button right there, uploaded data, Harpreet, on the very top. No, not there, on the very top. Okay, so maybe I should,
Okay.
let me quickly show you. Please follow along with me, Harpreet, so you can do it next time as well, okay? Okay, so whenever I want to check what was uploaded, I click right here. Okay, so this shows me that there was nothing that was uploaded, which means that it could be uploaded but unsuccessfully, right? So what does unsuccessful upload mean? Unsuccessful upload, and you can see that this is not loading, that's because that the process was not complete. So this visualization, this error, means that the upload was not done. So when you are creating your pipeline, it's always important to double check that the files were indeed uploaded. And so as Harpreet has shown, you have here the selection. First, you got to select the right selection. You have to complete the selection. When you click on File Upload, make sure that you're uploading the right file into the right place. Okay, great. And Courage is saying that his pipeline worked. So again, where do we see all of the pipelines that the participants in this group have run? We can go to the home of the organization page, and you will see the listing. And you can also see it under your own profile. So here, Courage has prepared this Ebola pipeline. Okay, I can see also Zuking Francis. I don't see anybody else. So hopefully other people have done it. So here, Ebola pipeline, right? Okay, perfect. So here we see. Okay, and you can see the variants right here, right? So if we zoom in, we can start looking at the variants. All right, great. Does anyone else have any other questions? All right, great. Thank you, everyone. We hope to see you again next time.